Happy Micro Monday. I'm going to demonstrate tomorrow's lab. This is going to be simple stains. So you're just getting your feet wet with the staining process. You're going to practice with 3 bacterial species. You have B. cereus, E. coli, and N. sicca. You're also going to practice using 3 stains: methylene blue, safranin, and crystal violet. For this lab, you're going to want gloves. You kind of have to have gloves for this one. You also may want goggles. I'm not going to wear mine because technically my glasses count as eye protection, and when I put them both on, it obscures my view a little bit. So the first thing you have to do for all staining is to heat fix, and that does 2 things. It kills the bacteria, and it adheres the bacteria to the glass slide, which helps a little bit and also hinders your view a little bit. I'll talk more about that when we get to the microscope part. You're also going to want a Bunsen burner. You need your striker. You need a loop. Right. I also recommend getting a bottle of DI water from our cart and one of these little beakers and putting just a little bit of DI water in that. I'll probably hand out your species. The stains are also on our cart. The glass slides are on our cart. And then the last thing you're going to need is the clothespin. This is our fancy slide holder. And then in our drawers, you also want to grab a staining tray, which is what we set our slides on. Okay, the first thing you have to do is light your Bunsen burner. You're going to hook it up to a gas jet. Mine's up here on the instructor bench. Yours is at your workspace. Just so you know, when you turn on these jets, 6 o'clock is full power. So this is on full power right now. If I keep going, it's going to turn it off. And I'm actually going to turn it at about 8 o'clock, 7 o'clock, somewhere in there, because I don't want it full power. A lot of times when you turn the gas on full power, it blows out your light. See how I did that? I squeeze this bar and it makes a spark. I hold it over the gas jet. I'll do it again for you. Got the gas going. Strike it, get it flame. So you can turn it down a little bit. We're using the Bunsen burner because your technique for staining will always be an aseptic technique, and that's to make sure the bacteria that you get on the slide is the one that you're supposed to be looking at, not like from your nostrils or your breath or something. Okay. So once you got your flame going, the way you do this aseptic technique is you hold your loop in that flame and see how it's glowing. That's how I know it's sterilized, because this loop has been sitting unused for a while. I'm going to do the whole thing. It's also good to do the entire loop when you get started, because it's really only as clean as the last person flamed it off. So I just go ahead and let that whole thing flame 2 or 3 times. Get it nice and hot, and that way it can cool down while I set up my slides. Okay, so I'm just going to set that down on the bench, and I'm going to prepare my slides. You have to do 3 species of bacteria and 3 different stains. And the easiest way to do this is to put 3 stains on a single slide. That way you can just cover the whole slide with 1 stain rather than trying to keep separate stains on the same slide. Make it a little bit easier. So I'm going to grab a Sharpie real quick. I'm going to label my slides since I'm putting more than one bacteria on them. So to do this, I'm just going to draw like mini fields of view to keep my bacteria smear separate. Got 3 circles. Do the same for the other 2 slides. And then you may want to label your slide of the species that you're going to use. For instance, maybe I like to keep them in alphabetical order, that way in case any labeling gets rubbed off, I'll always know they're in alpha order. And in the case of this lab, it's B. cereus, E. coli, and sicca. Sometimes the stain itself can wipe off your ink, so when I'm really feeling like being careful, I go ahead and do the back side at least with my circles. Just make sure the stain doesn't take it off when I rinse everything. I'm going to do the same for the back. The other thing we have going for us in terms of keeping everything straight on our slides, not only do I recommend keeping your bacteria in alpha order, but you can write on a piece of paper your labeling and just make sure your slides match your paper. And so I've done that for you on your lab helper down here where I talk about how to organize your slides. If you keep your slides in order that matches the lab helper, it doesn't really matter if your labeling comes off or not. You always know which is which. Okay. Labeling your slides is also going to help you right now when you get to the microscope part, because it's tough to learn how to find what you're looking for on the slide once you get it to the scope. And then using your permanent marker to give yourself a boundary will help you be able to find your bacterial smear a little bit more easily. Okay, I've got my slides labeled. I've got our flame going. I'm going to get started. So again, this is the heat fixing step, and this is like just getting the bacteria onto the slide. The second part is when you add the stain. But the first part of every staining process, no matter whether it's simple stains, gram stains, endospore, or acid fast, it always includes heat fixing. I'm going to go ahead and flame my loop again. You always want to do this right before you stick it into a culture tube, just to be sure it is nice and clean. And it's of course going to be about 1200 degrees Fahrenheit after you flame it and it glows. So you have to play a game between letting it cool before you stick it into your bacteria tube, but not letting it hang out in the air for too long. So one trick is once you get your tube open and your flamed loop ready to go, you can kind of hold it to the side of the tube and it'll cool off. You might have heard that sizzle. Now I know it's okay to go ahead and grab my bacteria. You can see it's in what's called a slant because I poured agar into this tube and then I let it coagulate at a slant, an angle. And I'm going to stick my loop in there and I'm just going to kind of swish it around, drag it up to get the bacteria off. Right. So that's how you get the bacteria out of the tube. I'm going to go ahead and flame my loop right now though, because I want to show you something before I smear my bacteria onto my slide. This is technically a wet mount procedure. So first we have to get a drop of water wherever you're going to lay down bacteria. And we have these squeeze bottles on our card that you can take back to your workspace. And you can either squeeze out, a drop of water into your labeled circles for where your bacteria is going. I find that difficult. I don't find that these bottles are real easy to control precisely. So what I do instead is, that's why I have you use these little 50 ml beakers. I put a little water in the bottom there, and I actually use my flamed loop. Doesn't matter how hot it is, I can stick it right in that water. I use that to pick up a little bit of water. And it's kind of like when you used to play with bubble wands. You can see when you've got the water in there, that water tension is tight enough that you can actually see it. And then once I have this, I'm going to go ahead and lay it first in my Be Serious slide 1. So I'm just kind of tapping it. Lay that down. The reason I'm being really careful about this water is that at the end of the heat fixing, we have to get all that water to evaporate. So the more water you lay down on a slide, the longer you have to wait for the drying. And remember, we're always going to be up against the clock with a 50-minute lab period. So anything you can do to minimize your inefficiencies will help you get through the labs. Okay, I'm going to reflame my loop because I'm going to go in and get my Be Serious. Okay, so I open the tube, kind of holding the cap and the tube with one hand. I go in, I'm going to lay it on the side, it's going to sizzle. I'm going to do the same thing, I'm just going to scrape around in there. Almost like you're doing a throat culture. And then I'm going to smear that bacteria into my water. I'm going to spread it out as thinly as possible. It's okay if you go outside your labeled field of view, that's not going to really matter. But you want a nice spread out thin smear. The thinner it is, the better it's going to be for you. If you lay it down too thick, aka if you see it with the naked eye on your slide, it's too thick, you need to spread it out. It's going to grab onto the stain really well and in layers that are going to obscure your view and just look really dark. You're not going to be able to see any detail. Also, as you spread it out, it's going to help it dry faster when we're moving in between the heat fixing stage and the staining stage. Okay, so I've got it smeared around. I'm going to put my Be Serious culture tube down, but before I do that, I have to flame the top part of the vial. Just kind of run it through the flame, 2, maybe like 3 times. And that creates a little air pressure, kind of like a vacuum, pulls all the air out of the tube to make sure it stays uncontaminated. And I'm going to flame my loop again. Get nice and glowing hot. And since I'm switching to E. coli next, I'm going to go ahead and flame the whole length of the wire on the loop to get a whole clean start. Okay. Get you a little closer. I'm going to get my water first. All right. I'm going to set it down in my E. coli space, back into the flame. Grab my E. coli, open it up. Try to keep the cap in a hand. Try not to set it down on the lab bench. Just in the interest of reducing contamination. Okay. And then I'm going to smear this around. And again, I'm going to spread it out a little bit. Flame my vial, cap on. back in the loop. Water. Slide. Flame. And by the way, the hottest part of the flame is down towards where the Bunsen burner starts. However, I think that's actually hotter than the glowing. So when you're going quickly, it helps to hold your loop up towards the top of the flame. If I hold it down here, it doesn't glow as quickly. So I like to keep it up here. Another thing that helps is to hold your vial horizontally at all times. That reduces the amount of air particles that get down into your vial while you have it open. Okay, this one's really thick, so I'm going to show you what a bad smear looks like. I won't pretend I did this on purpose for your benefit. Put my vial down. So you can see the B. cereus and the E. coli are pretty transparent. And I know that's going to be a really good smear. It's not going to pick up too much stain. It's not going to be obscuring to the view. The sicca, however, I put down way too thick. See how it's like milky? So that's going to be a little bit more problematic. Even the E. coli, you can see it's a little thick. Which doesn't surprise me because I had to spread it out. I'm going to move on though. So once you get all your smears, the heat fixing part really refers to what I'm about to do. You see I have a hold on my slide with my clothespin. The challenge of this is not setting your clothespin on fire. I'm just going to run my slide, not my clothespin, but my slide through the flame just a few times, really quickly. And that's going to heat up the slide enough to get that water to evaporate. And you don't want any water on your slide when you're staining. Hi, I'm making a video for Bio 200 right now. Okay, I'm just going to do the same two things for my second and third slide. This time I'm going to kind of do all my water at once. So I'm actually going to lay it down, not just on my 3 fields of view for the second slide, but I'm going to do it on all 6 fields of view for my second and third slides. Water really shouldn't evaporate, but if it does, you can always put down more. Okay, let's start with B. cereus. And again, I'm just going to go down in the tube, kind of scrape it, again, like you're doing a throat culture. Okay, get it on here once. I'm going to go ahead and lay it down on my third slide. Notice I didn't even have to flame it again because I got enough bacteria on my loop that I could go ahead and do it for the second and the third slides. It's probably best protocol if you flame between each one. Since Dr. Marshall's trying to have use the room, I'm going to be a little quicker than I would normally do. Okay. going to do my E. coli. Down in here. And when you get your culture tubes, you'll be able to see where the bacteria is. And you'll see the sort of translucent to milky growth. So you'll know where to go to grab your species. Okay, put that out. This one I'm actually going to flame in between. Laying down. Almost because I got too much on that last one. Okay, there's my E. coli. I'm going to go ahead and get my tube cap back on so it stays uncontaminated. You know, in that way, worst case scenario, if I do my stains and I look on the scope and it's just not a good stain, I can still go back to my vials and start over. I don't have to worry about my original cultures becoming contaminated. Okay, last one. And sicca. By the way, in sicca, very close cousin to gonorrhea, in the same genus. And in sicca tends to have like a yellowish color when we culture it. So it's, again, very easy to see in the tube usually. On, flame the loop so I can set it down, because you always want to make sure that it's cleaned off before you put it down in a shared space like the lab bench top. Just going to heat fix those last two slides, although I can tell the water's already started to evaporate, which is nice, but remember the other point of heat fixing is to kill the bacteria and help it adhere to the slippery surface of the glass slide. I'm being really quick when I put this in the flame, because if you hold it over it, it will crack. I've only had one student explode a slide in the flame, but I'd love for my stats to stay at 1. And let's see, they're all sitting on my stain tray. They're drying, and again, you can see where I've laid it on way too thick. So you probably won't believe me until you get yours on the scope for the first time. But absolutely, less is more when it comes to staining. So I'm going to at least start with my first stains. It looks like they're mostly dry, but I know that first one's really dry. I'm going to go ahead and turn my Bunsen burner off. Okay. And the reason I had you put your 3 bacterial species on 3 slides was, again, so you could treat them all with the same stain easily, since all 3 bacterial species you're going to want to try with all 3 stains. Since they're all together, I can just do 1 slide for methylene blue. I can do the second slide for another stain. I'm going to use crystal violet. And then the third one, I can use safranin. And to stain, you just squeeze the bottle to cover your smears completely. And then with most stains, there's a sitting period to allow it to work its way into the cell walls and cell membranes. For all of yours today, the methylene blue, the crystal violet, and safranin, they are all going to be 30 to 60 seconds. So it's not a big wait time. By the time I'm done with all 3, my methylene blue will probably be ready to rinse. But you can set a timer, you know, on your smartphone. You can watch our clock that's above the door. It has a second hand if you need. Okay. You don't have to be wasteful, but you do want to get complete coverage. Three species on one slide. It actually might be helpful to not do how I'm doing, where I'm just doing the whole slide. It might be helpful for you to make some breaks. So again, you know where to look once you get this on the microscope. So with the safranin here, I'm trying to be a little bit more careful. And maybe go back and cover that in sicca a little bit more completely. And now we're just going to wait 60 seconds. Again, probably ready to rinse my methylene blue since it's been sitting since the beginning. And for this, you're just going to use that DI water squeeze bottle and rinse off your slide. It's kind of hard to do one-handed like I'm doing with the video. So I'll put this down and show you. Probably have a little easier time of it if you go back to your trusty slide holder, and you can tilt it. Tilt it right into your staining tray. It's what it's there for. Just go ahead and rinse it off. Looks like I got a little in the back when I was being one-handed and sloppy. And you can see all the stain is rinsed off. You can see where the residue is. It's going to be really difficult for me on the microscope slide where I put the bacteria on there too thickly. Rinse. The cereus is really my best one here, and there's no residue, no thick smear of the bacteria, no big staining blotches, and I have a little stain that drips on my rack here that keeps interfering. Once you're done rinsing off the stain, you're going to use this pad of paper to further absorb any excess moisture and stains. It's called bibulous paper. I'm just going to give you a whole pad. And I'm going to lay that down in here, and then I can just kind of press gently. I'm just going to leave it closed while I rinse my second slide. Grab it with the holder, just rinse it off right into the pan. The back side just to make sure. There we go. And into the bibulous paper pad. Press it just a little bit. Last one. Get a good grip. I'm going to rinse off all this stain. This one looks a little better. And again, a really good stain you can't see with the naked eye, so you're probably going to be tempted to think you didn't get anything on there, but I can already see these two are going to be a little more challenging when I get them on the scope. Just because if I can see it with the naked eye, it means it's going to be really thick, hard to see through on the scope. And press. And now we're ready to take this to the microscope. So officially this is the staining process. It's done. And I'll make a separate video for the microscope just because you'll use that separately for all of the staining techniques. So see you next video.